my very great pleasure to introduce Professor Tim Elliott to you this evening. I've known Tim for over 30 years and I've admired his career for a long time. He started off as a biochemistry student here in Oxford, did his PhD in Southampton and then a postdoc at MIT, and came back to Oxford in the late 1980s to a Wellcome Trust Fellowship. Was appointed as Professor of Immunology in 2000, but shortly after that left for the University of Southampton, where he spent the last 21 years. He had several very successful projects there, particularly the Institute for Life Sciences. He was the brainchild behind that, a trans-university interdisciplinary grouping, and then latterly as the fundraiser and then director of the Centre for Cancer Immunology, and has directed his last 22 or so years towards the immunology of cancers. And in 2021, he was appointed as the Kidani, or the inaugural Kidani Chair for Immuno-Oncology, and he is the co-director of Oxford Cancer and the co-director of the COUK Centre. Of course, this is the Centre for Personalised Medicine lecture, and I have quite a personalised connection with Tim that some of you in the audience will know that, but for full disclosure, he does sometimes also answer to Mr. Lucassen. And despite 30 years in the same department, this is the first for me, so I'm quite nervous introducing him because neither one of us have ever introduced each other for a talk like this. But I think you will really enjoy this. We've promised an interactive talk. And I just want to say that our two daughters are very sorry to miss this lecture today. But over to you, Tim.
Great, thanks very much. And, you're nervous. Okay, so yeah, I'm Tim Elliott. I've never answered to Lucassen, although I probably would now if you were to call me that. So yeah, so I'm all those things, and I'm going to say a little bit today about how we're using immunology to treat cancer in new ways. I will show you that this can be a personalised approach, but I want to take a more general introduction. I think any talk on cancer research should start with a reminder of the global health burden of cancer worldwide. Now, do you think you could pass my specs so I can read the numbers from the screen? Well, I know that globally, 17 million new cases of cancer are diagnosed worldwide, and that leads to almost 10 million largely premature deaths from cancer a year globally. In the UK, the contribution from UK cases are 275,000 or more new cases of cancer diagnosed every year, and that results in almost 170,000 premature deaths. And when you look at the cancer types that are contributing to those figures, you'll see that breast cancer, prostate cancer, lung and bowel cancers account for the majority of those, with over 50,000 new cases diagnosed every year. That worked a minute ago. I need an AI head. Right. Okie doke.
It was working, wasn't it?
Yeah, I'm going to have to.
It's working. Okay.
You got a little glimpse into our domestic situation. Thank you. This is to remind us that cancer incidence is on the increase, and it's not just because as a population, we're getting older. This is not just a disease of the elderly, although that's incidence in elderly folk is, of course, a major health problem. This is just to remind us that even amongst the under 85s, cancer has now overtaken cardiovascular disease as a major cause of death. And that's despite a significant amount of research funding going into cancer research. These are some figures from an organization called the International Cancer Research Partnership, which is probably the only organization that tries to collate data from around the world on cancer funding. And it's only a sample, and it's probably just a minority sample of the amount of money that's spent annually on cancer research. That's well in excess of 4 billion. If you include research in the private sector, of course, that's tens of billions. I use this slide too to point out these interesting trends. So research funding into the basic biology of cancer has been gradually decreasing since 2006, when these figures started, and research into novel treatments is on the increase. This probably reflects the sort of shift in knowledge generation to knowledge application that we're seeing in more recent years. Nevertheless, the pipeline of new drugs for treating cancer, by the time those figures started, when research funding was at its peak in 2006, the new drugs pipeline, it was pretty much dry. There were no new drugs coming out into the market. That was, however, until immunology came along, when the number of new agents entering the pipeline began to shoot up as a result of some very exciting observations from basic immunology that were clearly exploitable in a cancer setting. And in 2015, cancer immunotherapy was declared the science breakthrough of the year. Immunotherapy is a completely new approach to treating cancer. If you're used to thinking of cancer treatments in terms of surgery and radiotherapy and chemotherapy, it's a living drug that involves biomolecules and living systems. And as such, by involving the immune system in attacking cancer, you have the opportunity to exploit some of the immune system's unique features as a biological system, including the ability to adapt to conditions inside the body and evolve an immune response over time. And the headline case for immunotherapy, of course, is that for the very first time, some very difficult to treat cancers like late-stage melanoma and non-small cell lung cancer actually showed cures in clinical trials. And as a result, some immunotherapeutic treatments are now first-line therapy for some of these very difficult to treat cancers. Okay, so let's dive into some biology. This is a public lecture, and so I'm not going to assume any prior knowledge. And I hope you enjoy the ride. So this is a picture of a small but aggressive late-stage melanoma. And if you look inside this small tumor and magnify about 1,000 times, what you'll see is an infiltration of white blood cells, lymphocytes. And some of these are cytotoxic T cells, and these are a kind of T cell that we think evolved to protect us against infection. This is a cancer cell, and it turns out that these tumor-infiltrating lymphocytes can discriminate between a cancer cell and an adjacent normal cell. So they can tell the difference between a cancer cell and a normal cell. And as a result of that, these cytotoxic T cells can become activated and engage in a cytolytic response against this target cell, destroying it. So if that was a virus-infected cell, the cytotoxic cell would kill it and prevent the dissemination of that viral infection throughout the body. If it's a cancer cell, it'll kill it and prevent it from dividing. So all the exciting bits of this cell-cell interaction, of course, is happening at this interface. So what's the cytotoxic T cells seeing or sensing on the surface of those cancer cells? And to answer that, I'll start by just taking you back to some school biology. Of course, proteins, every protein in the body arises from genes that are encoded in DNA, packed into nuclei. And this DNA can, throughout a lifetime, incur some mutations. And these mutations can quite often be translated into the corresponding protein, giving rise to an abnormal protein or a protein sort of encoding a non-natural amino acid at a particular position. And that can occur through environmental assaults like carcinogens, UV radiation, natural processes, just the natural process of poor fidelity of replication. Some of these changes are inherited changes, not many, and viruses can also have an impact on genomic DNA. So cytotoxic T cells can sense changes in normal proteins. How do they do that? Well, to start to understand how they do that, I'm going to play a little game with you. I'm going to hopefully illustrate this biological process through the medium of confectionery. And what you have to do, this is interactive, what you have to do is imagine that you're all cells in the body. You can be any cell, lung, skin, whatever. And I'm going to be a T cell on the lookout for some kind of change. My job is to detect those minor changes. So these chocolates each one of these chocolates represents one of those proteins derived from the translation of RNA encoded by one of these genes. And what I'm going to do now is distribute these throughout the cells in the body. So here's the interesting bit. There are more. What I'll do is okay. In the excitement there, something's happened, and I've just well. So hopefully most people have got one of these. So you've all caught your protein. What I'd like you to do now is to unwrap it. And when you've done that, just wave the foil wrapper in the air. Okay. Great stuff. You can, of course, eat the chocolate as a reward. Okay, so I'm a T cell, and I'm circulating around, scanning my tissues, and I see they're generally all vast, lots of nice gold rashes out there, and there's no reason to be alarmed. But I see down this corner we have something rather untoward. And that's an alert. That's an alert to T cell. I can act on that information. And I can go over and eliminate that rogue cell, which may be a precursor or an actual transformed cell. Okay, so let's just now think about the biological correlate of what you've just done. That's exactly how cells in our body deal with real proteins. When proteins reach the end of their natural life, they become slightly damaged, oxidized, partially unfolded, and they're targeted for degradation, which is that whole process of unwrapping the protein. And they're digested, they're turned over, just as you are doing now with the chocolates. But a bit of that protein, a fraction of all those proteins, are picked up by a hand. And that hand is actually a molecule called MHC class 1. That's a crystal structure of the molecule. And the only job of that protein, which is expressed on pretty much every cell type in our body, is to go to the cell surface and wave this little piece of the aberrant protein, or every protein in the body, for T cells to survey it. So we have here the molecular hand, the MHC molecule, and in this it's a beautiful little groove here that's fashioned out of 2 alpha helices and on top of a beta sheet, if we have any protein biochemists in the audience, a beautiful little groove like a hot dog bun with a short peptide like a hot dog sausage stuck in the middle of it, and that's our wrapper, and that's recognized by a specially adapted receptor on the surface of all our T cells that can specifically recognize this combination of our hand and a wrapper. And I've spent my whole life working that whole process out, and it's very gratifying to be able to see it enacted in this way. Okay, so we have now a molecular description of this recognition event of our lymphocytes recognizing the surface of a cancer cell. And it's, as I've said, an MHC molecule bound to a short peptide recognized by T cell receptor. So our immune system has a way of identifying these very slight molecular changes in cancer. And as I've said, that activates the T cells, and they can kill the tumor cells. So why then do we get cancer? Why is it such a problem? Well, to answer that question, we're going to play another game. We're going to switch it round. This time, you're going to be the T cells, and you're going to try and recognize the rogue melanoma cell. Before that, I should just let you know, remind myself, that the number of these lymphocytes that are infiltrated into a tumor do seem to be important in protecting us against tumor growth. So there's a really good correlation between the number of these cytotoxic T cells that you find in tumors and the survival, the overall survival of patients with those tumors. So this is data from a lab in Southampton, where that means tumor-infiltrating lymphocytes. In tumors with high levels of tumor infiltration, you can see that the overall survival is significantly better than tumors with low infiltration. So they are good, but they're not good enough to protect everyone against cancer. So why is that? So this time, you're the T cell, and you're going to try and pick out from a identity parade the melanoma cell from the normal cell. So as I've said, this tumor here is easy for us to identify at this sort of macro level, this ultrastructural level. It's easy for us to see the difference between tumor and skin. But as I've shown you, the immune system works at a molecular level. And at the molecular level, the differences just aren't that clear. So if we think about the molecular differences between normal and malignant, we think about normal as being we think about the cells as being a bag of Smarties in this game. And you can see there's not much difference between a normal cell and the cancer cell in terms of Smarties. I'll show you now how difficult that is for T cells. So I'm going to show you this identity parade of 18 cells. 17 of them are going to be normal. One of them is going to be malignant. And when you spot the difference, I just want you to put your hand up and keep it held up. Okay. You. Last. That game doesn't normally last that long. Okay, so we had 2 T cells recognizing the cancer cell there. And that was great. It's fantastic. But remember, this cancer cell is dividing all the time. And the 2 of you just could not keep up with a dividing tumor cell. You couldn't keep that cancer cell in check. As a result, we had a weak T cell response out there. Sorry, guys. And a cancer wins out. And that's why we get cancer, because by and large, cancer, the T cell response simply cannot keep up or is defective in some other way. But there are ways around that. And I just want to sort of explore one way that we're thinking of exploiting immunity to rectify that situation. And to do that, we're going to play the game again. Only this time, I'm going to give you a small clue. Okay? So when we play the game, I want you to play seriously. When you see the difference, put your hand up. Great. You see what happened? We had a prompt and a strong T cell response to that cancer cell. There was no hesitation at all. We all recognized the malignant cell. And that's what we've just done, is illustrated the principle of vaccination. So it might be possible, if we could identify the precise differences between normal and malignant proteins, then we might be able to isolate those differences and formulate them as vaccines and rehearse our immune system so that when that change actually occurs, we're ready. Okay? There is another way. So in that situation, of course, a strong T cell response is successful and the cancer loses. There is another way that we could have recruited more of you into that first round. Of course, we can't play it as a game, but if I'd have asked you who could spot the difference, then my guess is that everyone's hand would have gone up. There's one cell there that's just got lots and lots of green smarties. And so the abundance of a particular molecular difference expressed at the cell surface, we also think is important for recruiting T cells into that response. And that's something else that we might be able to exploit as a therapy. Okay, so now let's start talking about where these proteins come from. So, you know, what are the chocolates that we started out with? You know, do they have any characteristics in common? And the answer is yes. And this is the part of the talk where hopefully I'll get to personalization. So there are really 4 different categories of tumor antigens that define the difference between normal and abnormal to different degrees. And hopefully that will become obvious in the next 5 minutes or so. So the first category are viral proteins. And these are very attractive because they come from a completely, foreign, discrete genome that we know the sequence of it. And so if there are viral proteins that when they infect a normal cell drive transformation and are driving a cancer, then they might be highly specific for the abnormal cell and not present at all in normal cells. So they could potentially be strong targets for immunotherapy, for vaccination in this case. And they're also attractive because they are also candidates for prophylactic vaccination. And this is, this is out there. So vaccines against human papillomavirus have been available for, 15 years now. I remember when our girls were in the first cohort of vaccines. And all evidence points to these anti-HPV vaccines completely protecting against the development of cervical cancer. And there are other cancers that might also be driven by HPV. And so I think we can see rollout into other into the general population, certainly within my lifetime. Okay, so the second kind of tumor-associated antigen is kind of at the other end of the scale in terms of specificity. And these are proteins that are just overexpressed in the normal proteins that are present and expressed in adult tissue, but are just overexpressed in tumors. And a good example of that is this protein HER2/neu. It's a growth factor receptor, which is overexpressed in a lot of cancers, particularly breast cancer, and helps drive development and growth of those tumors. And it may be just like when we played the game earlier on, when I showed the array of Smarties. It may be that there are no T cells out there that recognize just a few of these molecules on normal cells, but when that difference is abundant at the cell surface, you might recruit in some of those T cells that can recognize this normal protein. The problem here, of course, is that it's also expressed on some normal cells, and so there's the problem potentially of toxicity. Okay, so the third group of tumor-associated antigens is really interesting. These are proteins that are important for normal embryonic development. So they're present in embryonic tissue, but aren't normally present in adult tissue, but get reawakened during the transformation process. And so the only adult cells that are expressing the protein are cancer cells. And these are attractive targets that have been used in vaccination studies, and I'll hopefully talk about those towards the end of my talk. And then the fourth category of tumor-specific antigens are the ones that have attracted a huge amount of attention more recently, and these are so-called neoantigens. So these are mutations that accumulate in cancer cells as the cancer develops, and can be highly individual. So different tumor types accumulate these point mutations, all sorts of mutations, but largely point mutations to different extents. So there are some highly mutagenic cancers like melanoma and lung cancer that can accumulate mutations at a rate of 10 to 100 somatic mutations per megabase of DNA, and others down at the other end of the spectrum, like some of the leukemias that are not very mutagenic at all, and don't look that different to normal myeloid or lymphoid tissue. So some of cancers up at this end of the spectrum, can have about 300 of their 30,000 or so cellular proteins bearing a mutation, which is remarkable when you think about it. And these mutations sort of accumulate over time and accumulate along a sort of evolutionary path. So if you're used to looking at a tree of evolution, this diagram will make some sense. That a mutation down here in a sort of a founder transformed cell will accumulate a new mutation that gives rise to a new branch of clones, and that will progress, and a further mutation will result in another branch, and so on and so on. And so mutations down that are very early are shared by a lot of the tumor cells, and those that occur later on in tumor evolution will only be expressed in a subset of tumor cells. And that evolutionary path is different for every single tumor. and maybe even every single metastasis arising from the same tumor in an individual. So what we end up with, if we go back to our Smarties, are a bunch of molecular changes that are either widespread, so-called truncal neoepitopes, because they refer to the sort of trunk of the evolutionary tree. And these are good targets for vaccine because they'll be shared amongst most of the tumor cells. Let's say this is diagnosis, slice across the tumor at this stage. Others will be only expressed in a subset of a large subset of cells, and some will be expressed right at the very end of these evolutionary branches and will only appear in a few cells inside a tumor. And these are therefore much less attractive as vaccine targets. The exciting thing is that some of these truncal mutations might also reflect mutations in genes that are involved in tumorigenesis, in driving the development of a cancer. So they might actually be shared between different individuals. And so this kind of neoepitope are extremely attractive targets for vaccination because they could be deployed in lots of different individuals using the same formulation. So what we can see is a situation in which we could develop different kinds of vaccines that target different or ever-decreasing populations of patients, from the very general vaccine that could potentially treat many different cancers across different cancer types even, by stimulating T cells that recognize these tumor-associated antigens, through to vaccines that might be deployable in groups of patients, perhaps patients with the same indication or with the same molecular driver type, these so-called shared neoepitopes that I just talked about, right the way down to personalized vaccines. So these are mutations that occur only in a single individual, a single patient. And that might sound like science fiction to some. It certainly did to me when the idea was first purported, but it's something that now with developed technology is possible and is reaching clinical trial. And so what happens is that patients at diagnosis, as their tumor sequenced, so usually exome sequencing, along with a little bit of associated normal tissue, and that allows us to identify these small differences between the normal and the tumor cell. And the differences are in the order of 1,000 to it could be tens of thousands of differences in nucleotide sequence, some of which will be coding, others not. The trick then is to predict which ones are likely to provoke a T cell response, and this is where all the kind of the computational magic is starting. And I'd have to say we're right at the beginning. We're just taking baby steps into this field of predicting which of these small mutational changes might give rise to peptides that could be presented by MHC molecules in a way that stimulates a T cell response that will eliminate the tumor. There are, however, a growing number of algorithms that are available that are able to shortlist that 10,000 down into tens or hundreds of potential candidates. And then after some sort of further computational and sometimes experimental grooming or filtering, these sequences can then be formulated into a vaccine. These can be either making a peptide itself, just the oligopeptide, synthetic oligopeptide, or probably more likely in the future, encoding these sequences into mRNA or even DNA or a viral vector that can be deployed as a vaccine. These vaccines are then given back to the same patient, and their response is monitored, and the information on the level of response and immunological response and clinical response to those vaccines are then fed back into this loop to improve our predictive algorithms. And this was just an excuse really to show my one sort of experimental slide. So this is the work we do. It's the biology of assembly of MHC molecules with antigenic peptides. We're interested in building models that will simulate the whole process, that will allow us to predict the levels of a particular peptide MHC complex that appear at the cell surface, with a view, of course, to understanding whether they are immunogenic or not. So this cartoon here sort of represents a very simple mathematical model that we developed. Represents about 15 years of work and 10 billion quid worth of research funding. But I also show it to remind us that it's not just the peptide or the genome that individualizes this whole process. So the fact that we all have different proteins and different mutations in those proteins is, of course, important. And the rate of supply of those peptides to the MHC cluster molecule in particular cells is also important, and will be, to a certain extent, individual. But the other thing is that those receptors, those MHC molecules, are highly polymorphic. So what that means is that there are many different variants of those proteins in the population, and each variant will select a slightly different repertoire of peptides to wave at the cell surface. In fact, each one of us expresses up to 6 different MHC molecules on our cell surface, and that means that if you take the whole combination, it's a pretty individual marker, actually, the combination, our tissue type, and it's why transplantation is such a tricky process, because finding an unrelated donor for organ transplantation is very, very difficult. And of course, what we want to do in tumor immunology is to provoke exactly the kind of immune response that leads to tissue graft rejection. So that's an element of personalization that we need to build into these predictive models. And the third factor is, and I had no time at all to describe the complexities of exactly how that foil wrapper makes it onto the hand. So all that fumbling around that you did at the beginning of my talk actually represents a complex biochemical pathway, and those fumbling fingers are sort of represented by a number of cofactors in this pathway, and one turns out to be extremely important enough to merit its own parameterization in this model. It turns out that this particular cofactor is called tapacin, that we've worked on for many years, assists different HLA alleles to different extents. So there's a highly personal combination of these cofactors with your MHC molecules that's important in predicting which peptides will be selected for presentation. And yeah, so the upshot, just like in the novel, the upshot of all this work is a simple mathematical formula that describes the number of peptide MHC complexes from a particular protein at the cell surface as a function of the supply of peptides to the endoplasmic reticulum, and the competition between those peptides for binding to MHC. The unbinding rates of peptide from MHC, so the strength of the interaction between the wrapper and your hand turns out to be very important, and the extent to which this whole process is assisted by these cofactors, basically how dexterous you are at unwrapping and waving the wrapper in the air. So what we can do then is incorporate some of this information into these predictive algorithms. And we can also take on board new experimental techniques, so techniques that, for example, involve the purification of the MHC peptide complex, so the hand and wrapper complex, extract the wrapper from a complex mix from a complex mix, and characterize exactly what the sequence of those peptides are that are being waved at the cell surface. That's sort of an experimental validation of these more computational approaches to the likelihood of a mutation being immunogenic. So in the last couple of minutes, I'm just going to show you some data that's out there on the efficacy of this approach. So this is an experiment that we did 25 years ago in a mouse model with a tumor antigen formulated into a DNA vaccine. And it's really just to show you that vaccines really do work. So they protect mice against the development of this particular cancer if you vaccinate them before they develop the cancer. So in a prophylactic setting, they're extremely effective. And they're pretty effective too if you deliver the vaccine in a therapeutic setting. So once the tumor is visible, if you then deploy vaccines, you can prolong survival significantly. This would translate into years if this were a human experiment. We've seen vaccines deployed in human cancers over the past 10 years or so in the form of these tumor-associated antigens. These are the oncofetal antigens that I talked about. This is the overexpressed antigen that I talked about. These are other tumor-associated proteins that are preferentially expressed in cancer cells compared to normal cells in melanoma, breast, colorectal, and lung cancer. And you can see that I don't need to go into the details, but there is some effect in all of these small clinical trials. The latest data from neoantigen vaccine trials, so these are personalized vaccines, so these are individuals, each of the individuals in these clinical trials have had a vaccine formulated from them that will only work in those tumors, or only designed to work in their own tumors, are looking reasonably promising. So there are no numbers really to refer to, only that I think, I should have memorized this. I think that 5 out of the 7 patients in this clinical trial showed some clinical response. All of them showed an immunological response, so they're all making T-cell responses to the vaccine. But 5 out of 7 showed some clinical response. And in this clinical trial down here, this is interesting because it shows that the occurrence of metastases post-vaccination is much lower than it was pre-vaccination. I think this is a very encouraging result. But it's not great. And the reason for that requires a little deeper insight into what's going on inside these tumors. So it's not just the cytotoxic T cells that I've led you to believe infiltrate tumors sort of uniquely. There are other immune cells that are attracted into the body of those tumors, and some of those immune cells are suppressive, and a great example of that are so-called regulatory or suppressor T cells, and you can see that they're labeled in red in this immunohistochemistry experiment. So you see lots of yellow CD8 positive cytotoxic T cells, but they co-localize with these suppressor T cells. And just to wake you up at the end of the talk, we're going to play one last game. For this, you have to be sat next to someone. So if it's possible, if you want to sort of split up a little bit, if you're just one seat away from the next person, and I want you to hold hands with your partner. So the person on the looking at the screen here, the person on the left-hand side of the partnership, you're the T cell, you're the anti-cancer T cell, the one that we've already played. And the person on the right-hand side of the partnership, you're using your left hand, you are these suppressor cells. And it's your job to inhibit your partner from putting their hand up when they see the difference. So so what you're doing now, or some of you are doing now, is illustrating the biology of what's happening inside these tumors. That the T cells are trying to put their hands up because they can see the green smarty clear as day, because we've gone through this before. But the suppressor cell in the partnership is trying to prevent them from putting their hand up. And this translates into real biology. This is an experiment. It's not ours, but we've done this many times. Again, it's an experiment, a preclinical experiment in experimental mice. This is an aggressive tumor that grows rapidly over kind of 20 days if unchecked. But if you deplete these regulatory T cells, these suppressor cells, and there are immunological tricks of doing this elimination, you can see that you now restore immunity to this tumor. These tumors grow for a little bit, but then they disappear. And you can show that that's because the CD8 positive T cells that have been recruited into those tumors are not being suppressed by regulatory T cells. Now, the way that we played that game, the hand-holding business, if you translate that into molecular interactions between molecules on the suppressor cell and the tumor cell, one of those interactions is between a molecule called PD-1 that's expressed on T cells, activated T cells, and its ligand, PD-L1, on the surface of suppressor cells and other cells in the tumor, including tumor cells themselves. And so you can see, straight away, a rationale for immunointervention here. If there's a way of preventing that interaction from happening, then you might be able to release the T cell from its suppression in situ and restore that immunity that we saw in the experimental model. And of course, that's exactly the basis of the immunotherapeutic treatments that have hit the news over the past 7 years or so, particularly this molecule anti-PD-1, which won a Nobel Prize for its discoverer, Jim Allison and Tasuku Honjo. So that pair is here. There are multiple inhibitory receptors that are present on T cells that are potential targets for immunotherapy in the same way. And most of these are now in clinical trials, and time will tell how effective they are. But the anti-PD-1 therapeutic, along with anti-CTLA-4, have been licensed for first-line therapy in cancers, either as monotherapies or, more often than not now, in combination with one another, because their action turns out to be synergistic. And this is just to show you what all the fuss was about around 10 years ago. This was the clinical trial that really shook the world. It was a clinical trial of very late stage melanoma. One arm of the trial was treated with this an antibody that blocked one of these inhibitory interactions, the CTLA-4 interaction. The other one was standard of care at the time. And there are two things to note. First, that these are the sort of clinical responses. They were really profound. Some of these cancers were very well advanced and essentially disappeared over a period of 17 to 150 weeks with periodic immunotherapy during that time. The real excitement, though, comes in these long-term follow-ups of these clinical trials. You can see that not only has survival sort of been increased soon after treatment, you can see that there's a minority of individuals here that are essentially cured of the disease. And these survival curves now go out to several years, and there's a real cohort of individuals that no longer in which the cancer has not recurred, and that's a new observation, completely new observation. So, I mean, just to re-express that, the excitement of immunotherapy is that if this is the sort of standard of care available, and this is the usual kind of result in terms of effects, clinical effects of a new small molecule, a new chemotoxic drug, you see some increase in survival, but at some point, most, if not all patients will relapse. And so long-term survival doesn't really reach above baseline. But we see now with immunotherapy some real durable responses. So it's our responsibility now as cancer immunologists to ensure that that baseline gets higher and higher with an increasing amount of research activity in this area. So I thought I'd just finish with a bit of recent data from Oxford, clearly combining this checkpoint blockade immunotherapy with a vaccination approach that boosts the number of T cells that are specific for cancer, the tumor, and increase the number of infiltrating T cells, even though they may be suppressed. If you combine that with checkpoint blockade, you might then release the inhibition on that increased population, and expect to see some synergy between vaccination and checkpoint blockade. And that's exactly what you see in these preclinical models. I don't want to go through all the data, just as this is a recent paper from Benoit van der Ende at the Ludwig in Oxford. And this very exciting preclinical data has now been translated into a clinical trial, which is recruiting as we speak. The vaccine targets in this case are these tumor-associated antigens, oncofetal antigens, they're called MAGE-A3 and NY-ESO. So watch this space. In place of the usual sort of credits, what I want to do is just draw your attention to all the great contributors to Oxford Cancer. Oxford has an incredible cancer research community. At the last count, we're about 900 cancer researchers all the way across the university and our associated hospitals. And these, I think I've captured the logos of most of those. If you see one missing, then come and let me know, and I'll add it to the slide next time I give this talk. So thank you very much.